So now we will look at Chapter 26, the urinary system. And we'll start with an introduction. So we'll introduce you to the organs and then the overall functions of the urinary system. So first, let's have a quick overview of the organs that belong to the urinary system, and then we'll look at these organs in more detail in subsequent sections. We start with the kidneys, which function in regulating blood composition, including the excretion of waste and the maintenance of normal fluid and electrolyte balance, which includes pH. And in this process, the kidneys produce a substance called urine, and then urine will leave the kidney and flow along the urinary tract. The next structure is the ureters, which are paired muscular tubes that carry urine from the kidney to the bladder. Then you have the urinary bladder, which is a muscular sac for the temporary storage of urine. And then you have the urethra, which is a muscular tube that carries urine from the bladder to the exterior of the body. So not as many different structures to talk about in this particular system. So some more terminology. Urination is also called micturition, and this is the process of eliminating urine from the body. So if we look at the flow of urine through the urinary system organs, urine is produced by the kidneys. It then flows out of the kidneys through the ureters, and then from the ureters it is going to flow into the bladder for temporary storage, and then from the bladder it will pass through the urethra to the exterior of the body. So the urinary system has three main functions. The first function is excretion. So the urinary system allows us to remove metabolic waste from our body fluids. So as blood passes through the kidney will filter the blood and remove waste and then add those waste to the urine. So the next step is elimination, and that is the discharge of the waste from the body into the environment. So that is the actual act of urination, in which you're taking the waste products that have been put into the urine and you're getting them out of the body. But also very important is homeostatic regulation. So the urinary system plays a large role in helping the body to maintain the appropriate blood volume and the solute concentration within the blood. So as part of the homeostatic regulation, the kidney helps to regulate blood volume and blood pressure, and it does this by adjusting the volume of water that is lost in the urine, and it also secretes two hormones, erythropoietin and renin. The kidney also helps regulate the plasma concentrations of ions, so it can adjust the quantities of sodium, potassium, chloride, and other ions in the urine. And then the kidney also secretes calcitriol, which is involved in regulating blood calcium levels. The kidney also helps to stabilize blood pH, so it can adjust the quantities of hydrogen ions and bicarbonate ions in the urine, and we'll cover that in more detail in Chapter 27. And the kidney helps to conserve valuable nutrients like amino acids and glucose, and then also to get rid of waste products like urea and uric acid. The kidney can also assist the liver in detoxifying drugs and poisons, and also the deamination of amino acids, which is taking the amino group off of the amino acids when the rest of the amino acid is needed to build other molecules. In this section, we'll look at the gross anatomy of the kidneys, so the macroscopic structures, starting with their position. So the kidneys are in the back of the abdominal pelvic cavity, and the left kidney is slightly more superior than the right kidney. And the adrenal glands sit on the superior surface of each kidney. And the kidneys are retroperitoneal. So if you look at the picture over here, remember the pancreas is also retroperitoneal. So these are all organs that sit behind the parietal peritoneal membrane that lines the peritoneal cavity. The kidneys are held in place by the overlying peritoneum. So again, they're behind the parietal peritoneum. They are also held in place by adjacent visceral organs and then supporting connective tissue that we'll look at the different layers of that in just a moment. So a floating kidney is the term that is used when you have a displacement of the kidney that results from either breaks or detachment of the connective tissue that holds it in place. And with a floating kidney, this can place stress on the attached vessels and the ureter that is leaving the kidney. So the kidney itself, if we look immediately around the kidney, there are three connective tissue layers that help to keep the kidney in place and also to help buffer it from shocks and jolts as we move around. The first is the outer fibrous capsule that surrounds each kidney. Then you have a layer of perinephric fat, which is a thick layer of adipose tissue around the capsule. And then you have the renal fascia, which is a dense fibrous layer that anchors the kidney in place by using collagen fibers. And again, to follow up on the idea of a floating kidney, here's what that would look like. So a floating kidney is also called nephroptosis. And so the danger with the floating kidney is it can bend the ureter and the renal vein and the renal artery into strained positions that can affect blood flow and urine production. So the fibrous capsule is the layer of collagen fibers that cover the entire organ. And in this top picture, you can see a little bit of the fibrous capsule that has been cut and peeled back. And then the hilum of the kidney is this prominent medial indentation. So you can see the hilum up here, and you can see the hilum down here. So the hilum is a point of entry for the renal artery and the renal nerves, and the renal nerves aren't shown in this picture, but you can see the renal artery in the top and in the bottom. So the renal artery is going into the kidney. The renal nerves would also go into the kidney. And then it's a point of exit for the renal vein, which will be coming out of the kidney, and also the ureter, which will be coming out of the kidney. So basically, four things are located at the hilum. So now let's take a look at the internal microscopic anatomy of the kidneys. So the kidney can be broadly divided into two areas. So the renal cortex would be the superficial outermost region that is in contact with the fibrous capsule that surrounds each kidney. The medulla then would be the deeper region that contains the pyramids. So in the renal medulla, we have renal pyramids, which are these distinct triangular shaped structures, and you typically have between 6 to 18 per kidney. And the base of each pyramid touches the cortex. Remember, we use the word base to describe the widest part of a structure, and apex is the more narrow part of the structure. So the base is out here where it touches the renal cortex. And then the apex of each pyramid ends in a structure called the renal papilla. So at the bottom here of each of these pyramids, at the pointy end, you would have a renal papilla. And then you have renal columns that separate each pyramid. So what we call a kidney lobe, and it's shown here by a dotted black line, a kidney lobe would be one renal pyramid, the cortex above the pyramid, and then portions of the adjacent renal columns on both sides. So when urine is produced, and we'll cover urine production in a subsequent section, when urine is produced, the papillary ducts discharge the urine out of the renal papilla, which is at the apex of each pyramid. So then that urine is going to be dumped out of the renal papilla, and it will be collected in a cup-shaped drain that is called a minor calyx. So these small areas right below the renal papilla would be a minor calyx. Then a major calyx forms from the merging together of 4 to 5 of the minor calyces. And then you have the renal pelvis, which forms from the merging together of 2 to 3 major calyces. and the renal pelvis will funnel the urine into the ureter. So if we look at the urine flow within the macroscopic structures of the kidney, the urine is going to come out from the renal papilla. It will then dump into a minor calyx. It will then dump into a major calyx. It will then collect in the renal pelvis and then pass out to the ureter. In this section, we're going to look at the kidney blood flow and innervation. So the kidneys receive about 20 to 25% of the total cardiac output, and blood flow through the kidney is at a rate of 1,200 milliliters per minute, so quite a lot of blood flowing through the kidney. So blood enters the kidney through the renal artery, so this is where oxygenated blood is coming into the kidney. The renal artery then divides into 5 segmental arteries. The segmental arteries further divide into interlobar arteries that pass through the renal columns between each pyramid. So you can think of the interlobar arteries as passing between one kidney lobe and another kidney lobe. The interlobar arteries then branch into arcuate arteries, which you can think of as arcing over the base of each renal pyramid. The arcuate arteries then supply blood out to the cortical radiate arteries, which you can think of as radiating out into the renal cortex. And then from the cortical radiate arteries, blood will then enter the nephron, which is the functional unit of the kidney, which we'll talk about in a subsequent section. So for now, we're just going to talk about the blood flow through the nephron. So again, we just left the cortical radiate artery, which is shown down here in the bottom image, and then blood will enter the nephron through what is called the afferent arteriole. And the afferent arteriole diameter can be changed to control blood flow to the kidney. Blood will then flow through the nephron capillaries shown here, which are called the glomerulus, and they are inside the glomerular capsule. This is the site where filtration actually happens, and we'll cover that in more detail later as well. And then blood exits the nephron through the efferent arterioles. So think of E for exits, and it exits through the efferent arterioles. The diameter of the efferent arterioles can also change to control kidney blood flow. And then the blood will then pass through a second set of capillaries that wraps all around the nephron, and these are called the peritubular capillaries. And this is where reabsorption and secretion are going to move additional substances between the blood and the urine. And then blood will leave the peritubular capillaries and flow into a venule. And as you can see from this picture, the venule will then dump into a cortical radiate vein. So the blood moving out of the kidneys mirrors the blood flow into the kidney, with the exception of the segmental arteries, because there are no segmental veins. So we just finished talking about blood flow through the nephron, and so blood leaves the nephron through the venule, and then it's going to drain into the cortical radiate veins, which again you can think of as radiating out into the renal cortex. The cortical radiate veins then drain blood into the arcuate veins, which again you can think of as arcing over the base of each renal pyramid. The arcuate veins then drain the blood into the interlobar veins, which pass through the renal columns between one kidney lobe and the next one. And then the interlobar veins will all join together to drain the blood into the renal vein, which is then going to take the deoxygenated blood out of the kidney. So if we review the blood flow, blood comes into the kidney through the renal artery, which then branches into segmental arteries, and then into interlobar arteries, which passes between the lobes and the renal columns, then into arcuate arteries, which arc over the base of the renal pyramids, then into cortical radiate arteries, which radiate out into the cortex, and then it's going to enter the nephron through the afferent arteriole, where it goes through the first set of capillaries called the glomerulus, where filtration happens. It leaves the glomerular capsule through the efferent arteriole, Then it wraps around the nephron in the paratubular capillaries where you have additional reabsorption and secretion. Then it's going to pass out of the nephron through the venule and then through the cortical radiate veins, which mirror the cortical radiate arteries, then through the arcuate veins, the interlobar veins, and then finally out through the renal vein. There are no segmental veins. And then if we look at the kidney innervation, the kidney is innervated by the renal nerves, and these contain mostly sympathetic nervous system fibers. Remember, the sympathetic nervous division is part of your fight or flight division, and so this is going to have the primary control over the kidneys. And it can adjust the rate of urine formation by changing the blood flow at the nephron, by changing the diameter of the afferent and efferent arterioles. The sympathetic nervous system can also exert control and influence urine composition by stimulating the release of renin by the kidney. In this section, we're going to look at the microscopic anatomy of the kidney, which includes the nephron, the juxtaglomerular complex, the collecting system, and then two types of nephrons. So as mentioned before, the basic functional unit of the kidney that produces urine is called the nephron. Each kidney has 1.25 million nephrons. That's 2.5 million with both kidneys. And they stretch out to a combined length of 85 miles. Each nephron is composed of a renal corpuscle and a renal tubule. So the renal corpuscle is made up of the glomerular capsule, which is also called Bowman's capsule, and a network of capillaries called the glomerulus. So here in this picture, this would be the renal corpuscle. And then it's also made up of a renal tubule, which is composed of a proximal convoluted tubule, a nephron loop, which is also called the loop of Henle, and a distal convoluted tubule. And then here is another image of what a nephron looks like, because a lot of times when they show you these pictures in textbooks, they have them all stretched out so that you can see the different sections, when in reality the nephron is more like it loops back on itself. So what I wanted you to see in this image is that the renal corpuscle is shown in green, and then the proximal convoluted tubule is shown in pink. Then you have your nephron loop or loop of Henle, and then what I wanted you to see is that the distal convoluted tubule actually wraps back around and comes very close to the renal corpuscle, and that'll become important in a little bit when we look at a specific structure. So then once each nephron creates some urine, that urine gets dumped into the collecting system, and we'll go over that in a minute as well. So let's take a closer look at these different parts, starting with the renal corpuscle. So the first part of the renal corpuscle is the glomerular capsule. The glomerular capsule has an outer wall shown here that is continuous with the renal tubule, and the first part of the renal tubule being the proximal convoluted tubule. So the glomerular capsule has this outer layer which makes up this outer wall, and then there is also a visceral layer, which are cells that wrap around the capillaries of the glomerulus. Between the outer layer and the visceral layer is the capsular space, and this is the space where filtrate from the blood is going to flow after it is filtered. Then you have the glomerulus, which is a knot of capillaries where the process of filtration occurs. Notice the size of the afferent arteriole and the efferent arteriole. So remember, the afferent arteriole takes blood into the glomerulus. The efferent arteriole drains blood out of the glomerulus, and the afferent arteriole normally has a larger diameter than the efferent arteriole, and that becomes important later on. So as mentioned earlier, the visceral layer of the capsule actually wraps around the capillaries that make up the glomerulus, and these cells that make up the visceral layer are called podocytes. Podo is a Latin word for foot, so these literally means foot cells, and they're so named because they have these little extensions of their cytoplasm which look a bit like feet, and in fact these extensions are called foot processes. And there are narrow gaps between these processes that are called filtration slits. So let's take a closer look at the filtration membrane, which is where the process of filtration occurs. And there are three components to the filtration membrane. First, you have the fenestrated epithelium of the glomerular capillary. So the capillary is actually a fenestrated capillary, which we covered in an earlier chapter. That means that the cells of the endothelium have these little pores in the walls. So that is the fenestrated endothelium. Then you have a basement membrane, which is also called the dense layer. And then you have those foot processes of the podocytes. So altogether, that makes up the filtration membrane. So during the process of filtration, which we'll go into more depth later, blood pressure is going to force water and solutes across this membrane. So it's going to go from the blood out into the capsular space that surrounds the glomerulus. There are also cells called intraglomerular mesangial cells, and here's one shown in this image over here. These are cells that are derived from smooth muscle, and they sit between the bends of the glomerular capillaries, so they can contract to decrease the diameter of the capillaries, so they can help adjust blood flow through the glomerulus. And they also have the ability to act as phagocytes. There's a condition called glomerulonephritis, which is inflammation of the glomeruli of the kidney. This is caused by immune complexes, and as a reminder, an immune complex is formed when an antibody binds to an antigen, and then other antibodies bind, and you get this large complex of antibodies and antigens all bound together. And so with this condition, those immune complexes clog up the filtration membrane, and that will impair the process of filtration. So this condition can be caused by any condition that leads to a massive immune response and the formation of a lot of those immune complexes. So there is also a structure near to the renal corpuscle called the Juxtaglomerular complex, or JGC. This is an endocrine structure in the kidney that is responsible for the release of the hormone called renin. So if you remember from previous chapters, the kidney releases a few hormones. Renin is one of them. Another hormone that the kidney releases is erythropoietin, or EPO. This one is produced by fibroblasts in the connective tissue of the kidney, and the kidney can also secrete calcitriol, which is produced by cells in the proximal convoluted tubule, and we talked about calcitriol a couple of times now when we've talked about blood calcium regulation. But the juxtaglomerular complex is responsible for the secretion of renin, and it is made up of three parts. The macula densa are tall, closely packed epithelial cells that are part of the distal convoluted tubule. So remember, I told you that the nephron actually has a bit of the distal convoluted tubule that wraps back around and goes next to the glomerulus. And so this is part of that DCT. And on one side, you'll find these tall, skinny cells, and they are called the macula densa. Then you also, and these cells function as chemoreceptors and baroreceptors. Then you also have modified smooth muscle cells in the walls of the afferent arteriole, and these are called juxtaglomerular cells. And these are the cells that are actually secreting the hormone called renin. They can also act as baroreceptors to monitor blood pressure in the afferent arteriole. And remember, the afferent arteriole is bringing blood into the glomerulus. Then you also have extraglomerular mesangial cells, which are shown here in this dark orange color. They sit in this little triangular space between the afferent and efferent arterioles and the loop of the distal convoluted tubule. These cells help to provide feedback between the other two areas, so between the macula densa and the juxtaglomerular cells. All right, so now let's move on to the renal tubule. Remember, the renal tubule is made up of three parts, and the first part is called the proximal convoluted tubule, or PCT. So this is the first section of the renal tubule. It is lined with microvilli on its cells to increase the amount of surface area that is available for reabsorption. And the primary function of the PCT is reabsorption of critical substances. And we're going to talk about these separate processes in making urine in an upcoming section. The next section of the renal tubule is called the nephron loop, or the loop of Henle. This is this U-shaped curve, U-shaped tube that connects the PCT and the DCT. And so it has both a descending limb, so a limb where the filtrate will be moving downward, and then an ascending limb on the upper side where the filtrate would be moving upwards. It has both thick and thin segments, and these are related to the type of epithelium that you would find in the different parts. So where you have squamous cells, it would be considered a thin part of the nephron loop. So there's a descending thin limb and a little bit of thin limb on the ascending side. And then with the thick ascending limb, you have actually cuboidal cells, so they're going to be a little bit thicker. So in the nephron loop, you have additional reabsorption that takes place, and the nephron loop also helps to set up a concentration gradient in the renal medulla, which we'll talk about in more detail later. Then you have the distal convoluted tubule, or the DCT, which is the final section of the renal tubule. This one has a smaller diameter, and the cells do not have any, if at all, microvilli. And as I mentioned before, the first part of the DCT actually wraps around and passes between the efferent and afferent arteriole. So most of the drawings you see in your textbook are like this one, where it's all laid out in a straight line, but in reality it looks more like this. And in these two images, the DCT is shown in the dark pink color and the PCT is shown in the light pink color. So again, you can see how the DCT wraps around and passes close by the glomerulus, which is where you have that juxtaglomerular complex. The primary function of the DCT is going to be secretion, although some reabsorption can occur here, but it is under hormonal control. And again, we're going to go into more detail later. So the next step after the nephron is that the urine will pass into the collecting system. So the first part of the collecting system is the collecting duct. So the collecting duct will collect the tubular fluid that comes out of multiple nephrons. So the collecting duct will begin in the renal cortex and descend down through the renal medulla. Some of the cells within the collecting duct do have microvilli, and the collecting duct can make final adjustments to urine before it passes out into the collecting structures that we talked about earlier, like the calyces and the renal pelvis, and then into the ureters. So the final adjustments that the collecting ducts can make is that they can help regulate the acid-base balance of blood by getting rid of either hydrogen ions or bicarbonate ions. They can also reabsorb water and secrete potassium ions when exposed to certain types of hormones that we'll talk about later. Then multiple collecting ducts are going to dump their urine into a papillary duct. So a papillary duct is collecting urine from multiple collecting ducts, and then papillary ducts empty into a minor calyx, as we mentioned earlier. So the urine flow within the microscopic structures of the kidney starts with filtration at the glomerulus. Your filtrate then passes into the proximal convoluted tubule, then into the nephron loop, then into the distal convoluted tubule, then into a collecting duct, and then a bunch of collecting ducts will go into a papillary duct. The papillary ducts will empty out of the renal pyramid through the renal papilla, and then the renal papilla dumps the urine into a minor calyx, and then that is going to pick up where we discussed the urine flow on an earlier slide. So here is a table from your textbook that is summarizing the organization of the nephron and the collecting system. So again, remember the nephron has two major parts, the renal corpuscle and the renal tubule. The renal corpuscle is made up of the capsule and then the glomerulus that is on the inside with the capsular space around it. And what is taking place here is the process of filtration. Then you have the renal tubule, which is made up of the PCT, the nephron loop, and the DCT. And the PCT is going to be primarily responsible for reabsorption, although there are small amounts of secretion that can happen in the PCT. The nephron loop is going to be responsible primarily for additional reabsorption, but it also helps to set up a concentration gradient in the renal medulla, which we're going to see later is very important for you being able to produce concentrated urine. Then you have the distal convoluted tubule, or the DCT, and here is going to be primarily secretion, but you can also have reabsorption when you have the presence of certain hormones. Then you have the collecting system, which is made up of the collecting duct and the papillary duct. And in the collecting system, the collecting duct will also be able to do some secretion and reabsorption, both under hormonal control. And then the papillary duct is going to be dumping the urine out into a minor calyx. So there are two major types of nephrons. The first type is called a cortical nephron, and with these, the majority of the nephron stays in the renal cortex. This is the majority of the nephrons, so 85% of all nephrons are cortical nephrons. They have a relatively short nephron loop that just barely dips down into the renal medulla. And then your other type of nephron that makes up the other 15% is called a juxtamedullary nephron, and these extend deep down into the renal medulla. So they have a very long nephron loop that extends deep down into the renal medulla. Because they have longer capillaries, their capillaries are actually called vasa recta, which is Latin for straight vessels, and they perform the same function as the peritubular capillaries that we discussed before, which are the name of the capillaries for the cortical nephrons. So now we'll introduce you to important terminology related to the formation of urine. So we'll talk about specific waste, and then the three major processes: filtration, reabsorption, and secretion, and then give you a final overview of urine formation before we talk about a little bit more detail. So one of the functions of urine is to rid the body of metabolic waste. And there are three important metabolic waste products that are eliminated by the urinary system. We've already talked about carbon dioxide and how that is released by the respiratory system, but the urinary system has different waste that it deals with. So urea is the most abundant organic waste. We generate approximately 21 grams of urea each day. And urea comes primarily from the breakdown of amino acids. And if you remember from the digestive system chapter, amino acids come from the breakdown of protein. So all that protein that you eat in your diet, a good portion of it is going to have the amino group stripped off of it in the process called deamination. And then that amino group is then combined with other amino groups to form urea. Then you have creatinine, which is a waste product from creatine metabolism. So if you remember from chapter 10, which we covered in A&P1, skeletal muscle tissue can store extra ATP, so extra energy, as creatine phosphate. And so in this metabolic process, you can get the formation of creatinine. And we generate about 1.8 grams of creatinine each day. And the rate at which creatinine is excreted can be used to measure kidney function, and they do that by a test called creatinine clearance test. The third waste product is uric acid, which is a waste product formed from the recycling of nitrogenous bases from RNA. And we generate approximately 480 milligrams of uric acid each day. And gout is a condition that occurs when uric acid crystals form within the synovial fluid of joints. It usually ends up affecting the big toe joint first of all. And these three waste products, uric acid, creatinine, and urea, can only be secreted in urine. So it is vital to have a healthy urinary system to be able to rid the body of these waste products. So the three processes involved in making urine starts with filtration. So with filtration, there is movement of fluid across a membrane that has pores in it, and the fluid is essentially pushed across the membrane by hydrostatic pressure, which in the body is blood pressure. So with filtration, it is the size of the pores that restrict which solutes can or cannot be filtered. So remember, in the kidney, in the nephron, we have the filtration membrane, which is made up of the endothelium from the capillary. Then you have the basement membrane, and then we have those little foot processes of the podocytes that have filtration slits in between them. So in the kidney, it's a combination of the pores in the fenestrated capillary and the filtration slits in between the podocytes that is going to determine what solutes can come out and which solutes are going to stay behind in the blood. So typically large proteins, things like red blood cells and white blood cells, are going to be too large and they'll stay in the blood. And then water and small solutes will be able to exit through the filtration membrane and become part of the filtrate. So filtrate, which is what is produced after the fluid has passed across the membrane, filtrate will contain water and only the solute small enough to pass through the pores. So here is an image, again, to give you an idea of what's going on. So blood is coming into the glomerulus through the afferent arteriole. Things that are small enough to cross the filtration membrane will be filtered out and will become part of the filtrate. Things that are too large, like the plasma proteins, will stay behind in the blood plasma. It's important to remember that filtration is non-selective, which means you can't pick and choose which solutes will cross the membrane. Anything that is small enough to pass through the pores will be filtered out of the blood. So this is going to include glucose, water, electrolytes, amino acids, waste products, etc. And so some of this stuff we're going to want to keep in our body, a good bit of the water, the amino acids, the glucose, a lot of the ions. And so remember that filtration is non-selective. Good and bad stuff gets filtered out of the blood. Because a lot of good stuff is going to be filtered out, we need to get that stuff back into the body. We don't want to lose it. And so that brings us to our next process, which is reabsorption. So when we talk about reabsorption, we are talking about taking solutes out of the filtrate and putting them back into the blood. So solutes are being moved from the tubular fluid in the nephron back into our peritubular capillaries, so it goes back into the bloodstream. So if you look at this picture here, again, we just talked about filtration, which takes place at the glomerulus. Then the next step is reabsorption, where we're going to take things that we want to keep out of the filtrate and we're going to put them back into the blood using those peritubular capillaries that wind all the way around the nephron. So reabsorption allows the body to keep important molecules that were filtered out non-selectively. So this is a selective process for solutes. It does require specific transport proteins for the specific solutes that we want to reabsorb. So for example, glucose is one of these. So there are glucose transporters that will take glucose out of the tubular fluid and move it back into our bloodstream inside the peritubular capillary. Water reabsorption is also very important. If we didn't reabsorb any of the water that was filtered out, we would become dehydrated in minutes. So much blood passes through the kidney and so much water gets automatically filtered out that we do have to reabsorb a good bit of the water. And water reabsorption happens passively by osmosis. Remember, water will always move towards an area of higher solute concentration. The next step is secretion. Secretion is essentially the opposite of reabsorption. So with secretion, we're taking specific solutes out of the bloodstream and putting them into the filtrate. So this time we are moving solutes from the paratubular capillaries into the tubular fluid. So again, if you look at the image, when we get down here to secretion, we are taking things that are still left behind in the bloodstream, again from those paratubular capillaries that wrap around the nephron, and we are transporting them back into the renal tubule to add them to the tubular fluid. Secretion lets the body get rid of additional waste products that were not filtered out during filtration. So again, remember, filtration is non-selective, and also if you have large things that can't cross through the pores, they're going to be left behind as well. So just because you filtered the blood out up here doesn't mean that everything you wanted to get rid of came out. So secretion is sort of a backup way of ridding the body of certain substances. And it's also a selective process for solutes, so it does require specific transport proteins for specific solutes, and an example in this case would be urea. Secretion can also be used to help adjust the blood concentration of certain ions by getting rid of extras. So if we have extra sodium ions, we can secrete those. If we have extra calcium ions, we can secrete those, etc. So overall, the process of urine formation is a combination of filtration, which is shown in this textbook image by the green arrow. So filtration takes place at the glomerulus. Then you also have water reabsorption and solute reabsorption. So the water reabsorption is shown by the blue arrows, and so you can see that when the blue arrow is going from the nephron back out to the outside, that would be reabsorption. And the difference between the dotted lines and the solid arrows is that in some places water reabsorption is purposeful, and in others it depends on certain circumstances. The same thing with the solute reabsorption. If you see the orange arrow that is coming out, that is purposeful reabsorption, and then the dotted orange arrows coming out represent reabsorption based on the presence or absence of certain hormones. So the arrows coming out represent reabsorption. The arrows that are pointing into the nephron would then represent secretion. So you can see that the secretion is always variable, so it's always shown by the dotted orange arrows. So this is a little bit complicated picture from your textbook, so I have another simpler picture to look at. But urine is the final product that you end up with after you have finished with the filtration, the reabsorption, and the secretion. So here is a more simplified diagram. So blood comes in the afferent arteriole to the glomerulus, out the efferent arteriole. So step number 1 takes place at the glomerulus, and that is filtration. Remember that when blood leaves the efferent through the efferent arteriole, it then becomes the paratubular capillaries. So step number 2, reabsorption, is going to be taking stuff from the tubular fluid and putting it back into the capillaries. Step number 3, secretion, is going to be taking whatever else in the blood you want to get rid of and putting it into the tubular fluid. And then when you're all done with that, you now have urine, and the final stage is then to excrete the urine from the body. So you can think of the excretion being what we do after we are done with the filtration, taking things away by reabsorbing them back into the blood, and then adding more things by secreting them from the blood into the tubular fluid. So in this section, we're going to go into more detail in the glomerular filtration and talk about filtration pressures, the glomerular filtration rate, and the regulation of the glomerular filtration rate. So as mentioned before, glomerular filtration takes place in the renal corpuscle across the filtration membrane. So as a quick reminder, the filtration membrane is made up of the fenestrated endothelium of the capillary, then you have a basement membrane, and then you have the foot processes of the podocytes with the filtration slits in between. The filtration that takes place at the glomerulus is a vital first step for all of the other kidney functions. So if you don't have the filtration happens, nothing down the line in the rest of the nephron can occur. So it is vital that filtration is maintained, and so that's why we're going to focus a little bit more on the filtration and the regulation of the filtration. So the driving force for the process of filtration is very similar to the way it worked in the capillaries, which we talked about in chapter 21, and that is that the driving force is hydrostatic pressure, which is your blood pressure, and this is taking place inside the glomerular capillaries. So the blood pressure, which is called the glomerular hydrostatic pressure, or GHP, when we're inside the glomerulus, this blood pressure is pushing fluid and solutes out across the filtration membrane. So there are some other pressures at play. For example, we can talk about the blood colloid osmotic pressure like we talked about back in chapter 21. That is the osmotic pressure caused by the plasma proteins that stay inside the blood. But that doesn't really change within the kidney. And then the colloid osmotic pressure should always be 0 in the glomerular capsule because none of the proteins get filtered out. They stay inside the blood. So that should always stay as 0. And there is a little bit of hydrostatic pressure that pushes back on the capillary from the fluid that does exist inside the capsule. But again, that's kind of negligible. So we're just going to focus on the hydrostatic pressure and the glomerular hydrostatic pressure, which is GHP. So there are other pressures at play, but the GHP is normally going to result in a net filtration pressure of 10 millimeters mercury that forces blood out of the capillary and into the glomerular space. And so again, remember, during filtration, we are forcing out water and solutes that are small enough to cross the filtration membrane, and they are coming out of the blood, and they're going into the capsular space that surrounds the capillaries. So let's see what determines what the glomerular hydrostatic pressure is. So the hydrostatic pressure in the glomerular capillaries is actually higher than in other systemic capillaries. So if you remember from chapter 21, the pressure inside the capillaries is normally around about 35 millimeters mercury, but here the glomerular hydrostatic pressure is 50 millimeters mercury. So it's higher than in the other systemic capillaries. And the reason for this is that the afferent arterial radius is larger than the efferent arterial radius. So if you imagine that you are taking fluid from a larger pipe and you are moving that fluid into a smaller pipe, that is going to increase pressure at the junction because you're going to have higher resistance from having this more narrow pipe over here. So having a larger afferent arterial than you have an efferent arterial bumps up the hydrostatic pressure within the glomerulus and makes that glomerular hydrostatic pressure higher than it would be in other systemic capillaries, and that is going to allow the filtration to occur at a more rapid rate. So the glomerular filtration rate, or GFR, is the amount of filtrate the kidneys produce each minute. And the glomerular filtration rate across both kidneys averages 125 milliliters per minute. So just stop and think about that for a minute. That means that if you didn't have reabsorption, you would lose 1 liter of body fluids every 8 minutes. So that's why I said earlier that reabsorption of water is a critical process that keeps us from becoming rapidly dehydrated. In fact, with this filtration rate, in one day, your kidneys make 180 liters of filtrate, which is pretty amazing when you think that you only have 4 to 5 liters of blood. So this shows you how much your blood is actually being filtered and then reabsorbed so that you don't lose a lot of water all at once. So the glomerular filtration rate, or GFR, is dependent on the net filtration pressure, which we just looked at as normally 10 millimeters mercury. And the net filtration pressure, as we just said, is driven mostly by the glomerular hydrostatic pressure, so the blood pressure within the glomerulus. Therefore, the kidneys are very sensitive to changes in blood pressure. Small changes in blood pressure can have a large effect on the blood pressure at the glomerulus, which can have a large effect on the filtration rate. So hemorrhaging and dehydration, both of which cause significant fluid loss, can quickly lead to acute renal failure. So because the glomerular filtration rate is so important to the rest of the function of the kidney, it has several layers of regulation. So first, let's look at autoregulation. Autoregulation would be changes in the local blood flow to maintain an adequate filtration rate. And this is accomplished through myogenic mechanisms, which is changes in the diameter of the afferent and efferent arterioles, as well as the glomerular capillaries themselves. So you covered this in your PhysioX in your laboratory course, but as a reminder, if the blood pressure drops, the afferent arterioles can compensate by dilating. So let's see how this works. So remember that the afferent arteriole is normally larger than the efferent arteriole, which causes an increased pressure, hydrostatic pressure at the glomerulus. However, if your blood pressure drops, there is also going to be a decrease in the glomerular hydrostatic pressure caused by the lower blood pressure. So we need to counteract this by increasing the pressure in the glomerulus if we want to keep the glomerular filtration rate constant. And so we can do this by dilating the afferent arteriole. This will increase the size difference between the vessel coming in and the vessel going out. So with a larger size difference, there's going to be increased resistance as the blood tries to flow from a very large vessel to a very small vessel, and that will increase the pressure inside the glomerulus back up, which will restore the glomerular filtration rate to normal. Also, an example from lab, if blood pressure rises, the kidney can compensate by constricting the afferent arterioles. And again, let's look at it. So again, normally, this is the normal condition. You have the afferent arteriole is slightly larger than the efferent arteriole. But if the blood pressure rises, then there will be an increase in the glomerular hydrostatic pressure inside the glomerulus. So to counteract that blood pressure increase, we want to decrease pressure here. And we can do that by constricting the afferent arteriole, which decreases the size difference between the vessel coming in and the vessel going out, which is going to lower the resistance and decrease the pressure in the glomerulus, which will restore the glomerular filtration rate back to normal. And in your PhysioX and lab, you also did some activities where you could change the efferent arterial diameter as well, but I'm not going to cover that in lecture. As far as lecture is concerned, I'm just going to use the afferent arteriole as an example. The next level of control is neural. And with the kidney, the neural control is provided by the autonomic nervous system. And as we mentioned earlier, it is primarily sympathetic innervation, which is your fight or flight division. So with the sympathetic signals, you will have powerful vasoconstriction of the afferent arterioles, and this will decrease the glomerular filtration rate. So again, if I have vasoconstriction of the afferent arteriole, I am making my two vessels closer in size, which is going to reduce the resistance, which will decrease the pressure in the glomerulus. And by decreasing the glomerular hydrostatic pressure, I will decrease the glomerular filtration rate. So in a crisis, like for example, if you had a sudden decrease in blood pressure from a heart attack, remember that if I have a decrease in blood pressure, I would normally want to increase my GFR, but this is an emergency and I don't necessarily want to be busy making urine when I'm having a heart attack. So the sympathetic innervation overrides those local controls. So again, normally with a decrease in blood pressure, I would increase my GFR, but when the sympathetic nervous system gets involved because of an emergency, it will counteract those local controls and it will cause vasoconstriction instead, and this will decrease the GFR. After the crisis has passed and the sympathetic tone returns to normal, the GFR will increase back to normal again. So basically, think of it as the sympathetic nervous system is diverting blood flow away from the kidney during the sympathetic activation. So you're in a crisis, it's your fight or flight system, you don't want to be busy making urine, so you're going to not have as high of a blood pressure inside the glomerulus, so you're not making as much urine. Now, this can cause a problem for endurance athletes like marathon runners or ultramarathon runners. So when you exercise, and especially when you do strenuous exercise, you do activate your sympathetic nervous division. So these people, the longer that they go on with their exercise, can end up having reduced kidney function, which can cause a buildup of metabolic waste in their bodies. And after a really long marathon or ultramarathon, athletes can even have proteinuria or hematuria. And proteinuria is when you have protein in the urine, which is an abnormal condition. Normally, you should not have protein in the urine. The protein should be too large to cross the filtration membrane. So this usually indicates damage to the filtration membrane. And hematuria is blood in the urine, which again indicates that the kidney is under severe stress. So some marathon and ultramarathon runners can even go into acute renal failure. And again, the underlying cause is having a extended period of sympathetic nervous system activation. So now we'll look at the hormonal control, and we've got two sets of hormones to talk about, but hopefully this should be mostly review for you because these were covered in previous chapters. So first we have the renin-angiotensin-aldosterone system, or RAAS, and the ultimate goal of this system is to restrict water and salt loss and help to increase the GFR to normal. So during this process, renin is released by the juxtaglomerular complex, and the release of renin happens under three different conditions. It happens when there is a decrease in blood pressure that is detected by those baroreceptors in the juxtaglomerular complex. This could be a widespread decrease in blood pressure, say for example caused by like hemorrhaging, or it could even be a decrease in blood pressure caused by a blockage of the renal artery. Also, you can have direct stimulation of the juxtaglomerular complex by the sympathetic nervous system, which can cause renin to also be released. And you can have a decrease in the osmotic concentration of tubular fluid in the distal convoluted tubule, which is sensed by the chemoreceptors in the macula densa. So once renin is released, it will convert angiotensinogen, which is an inactive form of a protein, into angiotensin 1, which is also inactive. And as a reminder, angiotensinogen is a plasma protein produced by the liver, so it will just be present in the plasma. So in the lung capillaries, an enzyme called angiotensin converting enzyme, or ACE, will convert the inactive angiotensin 1 to the active angiotensin 2. And then angiotensin 2 will act at the nephron, the adrenal glands, and the central nervous system to stimulate thirst, so you drink more water, to cause vasoconstriction of the arterioles and the precapillary sphincters, which will increase blood pressure, to promote antidiuretic hormone secretion, which will stimulate water reabsorption in the kidney that we're going to look at later, and to promote aldosterone secretion, which increases sodium reabsorption in the kidney, which we'll look at later. And the ultimate result is a restoration of a normal glomerular filtration rate, increase in blood pressure, and an increase in blood volume. So here is the picture from your textbook. This is showing a summary of the response to a decrease in GFR. So our homeostasis set point would be a normal glomerular filtration rate. So the stimulus would be a disturbance in that normal GFR, and in this case it would be a decrease in GFR, which for example could be caused by a decrease in blood flow to kidneys, which could be caused for example by a decrease in blood pressure. So this will end up having a decreased filtration pressure, which means we're going to decrease the amount of filtrate and urine that we're producing. So the endocrine response is to have the JGC release renin. Renin will trigger the formation of angiotensin 1, which then gets converted to angiotensin 2. We'll also have angiotensin 2 acting on the nervous system, which is going to stimulate the thirst centers in the hypothalamus to make you increase your fluid consumption. Also increase the ADH production, which will increase your fluid retention. And the activation of the nervous system will trigger the sympathetic system to cause constriction of systemic veins, increased cardiac output, and peripheral vasoconstriction, which will increase blood pressure. Angiotensin 2 will also have the adrenal glands make more aldosterone. Aldosterone will increase sodium retention, and with the sodium retention and the fluid retention, I'm going to increase my blood volume. And then angiotensin 2 will also constrict peripheral arterioles and further constrict the efferent arterioles. So altogether, I'm going to end up increasing blood flow to the kidneys and increasing my glomerular filtration rate, which brings me back to my normal GFR. And then the flip side, on the other side, what do we do if GFR increases too much? And that would be the natriuretic peptides, and we talked about these previously, atrial natriuretic peptide and brain natriuretic peptide. These are both secreted by the heart when the blood volume rises, which would also cause an increase in blood pressure, which would cause an increase in the GFR. And the ultimate goal of the natriuretic peptides is to promote fluid loss so that we can return our blood volume and blood pressure back to normal. So the natriuretic peptides will trigger the dilation of the afferent arterioles and the constriction of the efferent arterioles. And again, we're just going to focus on the afferent arteriole. So natriuretic peptide would trigger the dilation of that afferent arteriole. So we're making the size difference between the two arterioles larger, which is going to increase pressure in the glomerulus, which will increase the GFR. This will help us produce more filtrate and more urine, which will promote fluid loss. So this further increases the GFR. I have more filtrate collection, so more urine is produced, and that's going to help me promote fluid loss. I'm also going to have a decrease in sodium reabsorption by the kidney. So to summarize, the two major actions of the natriuretic peptide are to increase the filtration rate by dilating the afferent arteriole and to suppress the reabsorption of the sodium ions in the collecting duct and other areas so that I increase my secretion of water and sodium. And so my ultimate result is have an increased urine output and a decrease in blood pressure and a decrease in blood volume. So in this section, we're going to look in more detail at the renal tubule and also the collecting system, and we'll be looking at reabsorption and secretion in these areas. So the essential functions of the renal tubule include reabsorbing all of the useful organic nutrients that came out in the filtrate, and this is going to include useful substances such as glucose, free fatty acids, amino acids, vitamins, and other solutes. The renal tubule is also responsible for reabsorbing more than 90% of the water in the filtrate and secreting any waste that did not pass into the filtrate at the glomerulus, because remember, the filtration step is non-selective. So again, to summarize, in the process of urine formation, we talked about glomerular filtration in the previous section. So in this section, we're going to focus on the tubular reabsorption and the tubular secretion. So also note that there are a lot of cellular transport mechanism details covered in this chapter that I will not be covering. So make sure to use the learning objectives when you are studying. I do want to go over the general idea of transport that takes place in the kidney. So reabsorption and secretion, which we're talking about in this section, occur by diffusion, osmosis, leak channels, and carrier-mediated transport. We've talked a lot about diffusion, osmosis, and leak channels in previous chapters, both in A&P 1 and A&P 2. So I just want to go over a little bit more about carrier-mediated transport here. So carrier-mediated transport includes facilitated diffusion, which is where you have diffusion from a high concentration to a low concentration, but it takes place through a protein in the membrane. So facilitated diffusion means it requires a protein carrier. There is also active transport, which is going to move substances against their concentration gradient. So from an area of low concentration to an area of high concentration. And since things are being pumped uphill, so to speak, this process does require ATP. And then co-transport involves the use of ATP to pump one molecule against its gradient, like sodium in this picture, and then the molecule that was pumped against its gradient then diffuses down its gradient, and that energy is used to propel another solute. And so that is called co-transport, and in co-transport they both move in the same direction. And then there is countertransport, where again you're using ATP to pump one of two molecules across the membrane, and then when that molecule diffuses back down, it brings another molecule with it. But in the case of countertransport, the two molecules are moving in opposite directions. So some characteristics of carrier-mediated transport are that proteins are specific for the type of solute they allow to pass. So, for example, sodium channels will not allow potassium ions to pass, and vice versa. Proteins typically work in one direction only, so a glucose transporter that moves glucose out of the tubular fluid is not going to put glucose back into the tubular fluid. And the distribution of carrier proteins can vary in different areas of the cell. So the apical side of a cell can have a different complement of proteins than the basal side of the same cell. And one cell can contain many different carrier proteins. So one cell could have proteins for sodium, potassium, glucose, chloride, and a bunch of other stuff. Also, carrier proteins can become saturated, which means they are working as fast as they can, and this is the maximum transport rate for that particular carrier protein, and we'll see how this relates to glucose on the next slide. All of this was covered back in Chapter 3 of A&P 1, so you can always go back to review this information if you've forgotten. Let's look at glucose transport as an example of these principles. So, for example, if you have a protein called a glucose carrier, it will only transport glucose. It will not transport something else. Glucose will only be transported in one direction through the protein carriers. Different glucose carriers may be on the apical surface of a cell versus the basolateral surface. And this is an example picture that I showed you earlier, where we're taking glucose out of the tubular fluid and putting it into the bloodstream at the peritubular capillary. So I may have one type of glucose transporter here on the apical surface of the cells that are lining the tubule of the kidney, and I may have a different type of glucose transporter on the basolateral surface that is putting the glucose from the cell into the bloodstream. Again, your textbook goes into a lot of details, like it's secondary active transport on one side, facilitated diffusion on the other. We're not going to go into that level of detail. I just want you to be familiar with the concepts of how all of these transporters work. And now let's look at the concept of saturation. So all of these different molecules that are transported in and out of the tubular fluid in the kidney have what is called a renal threshold, which is the maximum rate at which that they can be transported either from the tubular fluid into the blood or vice versa. So for example, when your blood glucose levels exceed 180 milligrams per deciliter, there is so much glucose in the filtrate that the glucose carriers become saturated and they cannot transport all of the glucose out of the tubular fluid. So any blood glucose level in excess of 180 milligrams per deciliter is going to mean those carriers become saturated and they're not going to be able to reabsorb all of that glucose from the filtrate. And so again, normal glucose handling when you have normal blood sugar levels is that you are able to reabsorb 100% of the glucose out of the filtrate and back into the blood. But then if you have high blood glucose levels, then you're going to saturate those protein carriers. They're not going to be able to reabsorb all of the glucose, and you're going to be left with glucose in the filtrate. So as a result, glucose will get passed in the urine, and in fact, glucose in the urine is a key sign of diabetes mellitus, and we covered this disorder in a previous chapter. So now let's look at the reabsorption and secretion by segment of the renal tubule, starting with the proximal convoluted tubule. So this is the first place the filtrate goes after it has been filtered at the glomerulus. So overall, the proximal convoluted tubule is responsible for reabsorption of 60 to 70% of the filtrate. So there are 5 major functions that occur in the PCT. First, we have reabsorption of organic nutrients. So 99% of glucose, amino acids, fatty acids, and other important organic nutrients are reabsorbed. We also have active reabsorption of ions, and since this is an active process, it will use ion pumps that will require ATP. So in the proximal convoluted tubule, we're going to have a lot of reabsorption of sodium and potassium ions. We'll also have reabsorption of bicarbonate ions, also magnesium, phosphate, and sulfate ions. The PCT is also a place where you'll get some passive reabsorption of ions. So this is ions coming out of the tubular fluid and back into the bloodstream by diffusion. So they're just moving from an area of high concentration to low concentration, so no energy is required. And this is going to include things like urea, chloride ions, and lipid-soluble solutes. So I know urea is one of the waste products that we want to get rid of, but in the PCT, it is able to move by diffusion, so it can leave the tubular fluid and go back into the bloodstream, but then later on, we can actively secrete it to get it back into the urine. There is also a lot of reabsorption of water that occurs in the PCT, and this occurs by the process of osmosis. So as you have the reabsorption of all of these ions and organic nutrients, that is shifting the concentration of solute away from the inside of the PCT and into the bloodstream. And so water is going to follow the high solute concentration by osmosis. So as you're taking out all of these solutes, water will also come out and follow those solutes into the bloodstream. So remember, we have an extremely large amount of filtrate that we make every day, and so the PCT is actually responsible for reabsorbing 108 liters of water every day. And then a very small amount of secretion takes place in the PCT, and this is mainly hydrogen ions. So remember, the primary function of the PCT is going to be primarily reabsorption. So now we'll look at the nephron loop, and as a reminder, the nephron loop has a descending limb and an ascending limb, and the reabsorption and secretion differ between the two limbs. The nephron loop is responsible for reabsorbing the half of the remaining water. So out of all of the water that did not get reabsorbed in the PCT, half of what is left will be reabsorbed by the nephron loop. And the nephron loop is also going to reabsorb 2/3 of whatever sodium and chloride make it this far that weren't reabsorbed in the PCT. So in the descending limb of the nephron loop, this limb is freely permeable to water. So you have a lot of reabsorption of water, but it is not permeable to solutes. So only water is being reabsorbed in the descending limb. In the ascending limb, this side is not permeable to water, so there is no more water reabsorption on the ascending side, but you do have active transport of sodium and chloride ions out of the tubular fluid and into the blood, and this requires ATP because it is active transport. And the difference in permeability between the descending and ascending limb is the key to the creation of a concentration gradient that will allow concentrated urine to be made, as we'll see in the next section. So moving on to the distal convoluted tubule. So by this point, we have reabsorbed a lot of stuff, and only about 15 to 20% of what we initially filtered out at the glomerulus is left. So just a small volume is left, and now we are in the DCT. So the DCT is going to make final adjustments to the solute composition and the volume of the tubular fluid. So you do have some selective reabsorption of ions in the DCT. So there is some active reabsorption of sodium and chloride ions. Additional sodium can be reabsorbed in the distal convoluted tubule when aldosterone is present. In both of these cases, both when they're pumping sodium and when aldosterone is present and additional sodium is being reabsorbed, in both cases, the sodium coming back into the bloodstream is swapped for a potassium ion, so potassium ends up being secreted, 1 potassium for every sodium that the body conserves. So keep in mind that sodium conservation is linked with potassium loss. You can also have some calcium ion reabsorption if you have parathyroid hormone present, and we covered blood calcium regulation last semester and earlier this semester. So if you remember, parathyroid hormone tells the kidney to reabsorb more calcium ions, and that takes place in the distal convoluted tubule. But in the distal convoluted tubule, you also have a lot of secretion of ions. So you can secrete potassium to recover sodium, as we discussed up here. You also can secrete hydrogen ions to regulate blood pH, which we're going to talk about in the next chapter. You have some selective reabsorption of water in the DCT, but only if you have the hormone antidiuretic hormone available. So if there is antidiuretic hormone around, then you will also get water reabsorption in the DCT. Now let's look at reabsorption and secretion in the collecting system. So remember that the collecting duct is receiving tubular fluid from multiple nephrons. And in the collecting system, we can get some additional reabsorption. So we can get the reabsorption of more sodium, but again, it's traded for potassium. So for every sodium reabsorbed, 1 potassium is secreted. We can also get some reabsorption of urea and hydrogen and bicarbonate ions, which help to control pH. We can also have some secretion in the collecting system. We can secrete hydrogen and bicarbonate ions to control pH. And again, we're going to talk more about pH control in the next chapter. And you might wonder, well, how can the same structure both secrete and reabsorb both of the same types of ions? And if you remember when we talked about the different types of cells in the collecting duct, I mentioned that some of the cells have microvilli on them. These are called intercalated cells. And the collecting duct actually has two types of intercalated cells. So there is a type A cell, which has the proteins for secreting hydrogen ions and reabsorbing bicarbonate ions. And you have type B cells, which can secrete bicarbonate ions and reabsorb hydrogen ions. So which cell you activate is going to depend on the current pH situation and in which direction you need to change the pH. And again, we're going to go into that level of detail in the next chapter. In the collecting system, you can also get the reabsorption of water, but only if you have antidiuretic hormone present. And you can get additional reabsorption of sodium and secretion of potassium, again, only if you have a hormone present, and that hormone is aldosterone. So here's a nice little table to show you what is typically reabsorbed. So the ions and metabolites the body wants to keep, things that are typically secreted, so secreted ions, waste products, and then miscellaneous things like drugs and excess neurotransmitters. And then there are some things that there are no specific transport mechanisms for. They just diffuse in and out based on concentration gradients. And so here are the normal laboratory values for solutes in the plasma on the left side. So this is if you were to have a blood test. This is what your concentration should be in your blood on the left side. And then if you have a urine test, this is what the concentration should be in the urine on the right side. So you can tell, for example, with the metabolites and nutrients especially, like glucose can be between 70 and 110 milligrams per deciliter in your blood, but you really don't want any glucose in your urine. So the very, very small number in the urine means that we should be reabsorbing most of the glucose. Same thing with lipids, most of them should be reabsorbed. Most amino acids should be reabsorbed. Under normal conditions, there should be no protein in the blood. And then the waste down here at the bottom are the opposite. If you look at the value of the waste products in the blood, they should be much, much lower than the value of the waste products in the urine, because the purpose of the urinary system is to get rid of these wastes. Now with the ions, you can have a little bit more overlap because they can change based on different conditions. So the two values are not that different except for the potassium values. So in this section, we'll see how the countercurrent multiplication works, and we'll talk about the types of water reabsorption and the regulation of urine volume and concentration. So countercurrent multiplication occurs when you have active transport between two limbs of a loop in which the effect of the transport increases, so you get positive feedback that maintains a certain condition. And so you essentially have exchange between fluids that are moving in opposite directions. And we see this in the nephron loop. So remember that with the descending loop, we have permeability to water but not solutes. And then in the ascending loop, we have permeability to solutes but not water. And so we have fluid coming down the descending loop and then going back up the ascending loop. And this process allows for the formation of a concentration gradient. So let's see how that works. So let's start by looking at the ascending limb first. So we'll start with the concentration at the bottom of the nephron loop, which is 1200 milliosmolars per liter. And so as you are going up the ascending limb of the nephron loop, water cannot cross the membrane, but you do get active transport of sodium and chloride ions. So as you move up the nephron loop, these little transporters are pumping out the sodium and chloride, which is slowly decreasing the concentration left inside the nephron loop. So you can see we start at 1200 and we go up to 100. And because the transporters have more ions they can work with at the bottom of the loop, they end up pumping out more sodium and chloride at the bottom, which causes a higher concentration in the renal medulla outside of the loop. Now if we come over here and look at the descending limb, remember the descending limb is permeable to water, but not to solutes. So water can move, and water will move by osmosis to area of higher solute concentration. Well, now I've got this concentration gradient in the renal medulla that was set up by the pumping out of the sodium chloride ions. So at the top, water will diffuse out by osmosis until the two concentrations match. And then as you move down the descending loop, more water will come out as it gets more concentrated on the outside. You continue to come down, more water will come out again until it equalizes concentration, until you eventually get with a very concentrated solution at the bottom of the loop. So you have the max concentration at the bottom of the nephron loop. Now, because this can be a little challenging to understand, I tried to make a little illustration here to let you see what exactly is taking place. And it's easier to actually start at the bottom of the loop and sort of work our way backwards. So remember that at the bottom of the loop, we have a very high solute to water concentration. So high solute in relation to a low amount of water. And so as we come up the ascending loop, we have pumps that pump solute out of the loop and into the surrounding tissues. And at the very bottom of the ascending limb, we have a very high transport rate because we have such a large volume of solute. So as the solute gets pumped out, the amount of solute inside is going to decrease because it's being pumped out, and you're going to be pumping out a lot of solute down here at the bottom because you had a lot of solute to start with. So as you continue to move up the ascending limb, the rate of transport of the solute is going to decrease because I have a smaller volume of solute, so I will only have a medium transport rate up here compared to the high transport rate down here, just because I don't have as much solute left inside at this point. But I'm still pumping out solute, so at this point I'm pumping out more solute. I'm going to be left with medium solute behind in the tube, and I'm going to now be pumping out medium levels of solute into the surrounding tissues. And then again, as I continue upwards, I'm still pumping out solute, but at the very top I'm going to have a slower transport of solute because now there is a much smaller volume of solute left inside the nephron loop, so there's not as much to transport. But nevertheless, I am still transporting some out, and so I'll end up with low solute concentrations on the inside, and because I'm not pumping out as much, I'm going to have lower solute concentrations on the outside. So notice that the amount of water didn't change because the ascending limb is impermeable to water. So the only thing that is changing is the solute concentration. Now let's look at some actual numbers. And I want you to keep in mind that the numbers I'm showing and the numbers in your textbook are concentrations. So it's actually a measurement of solute per volume. And so these numbers are in units of milliosmoles per liter. So at the very bottom of the nephron loop, we have a concentration of 1200. So as I'm moving up, I am reducing the solute concentration inside the nephron loop. So it's going to go down. But I'm also pumping a lot of the solute out, so I'm going to have a high solute concentration out here in the tissues. And then as I move up, I'm going to get lower and lower inside the tube, and I'm going to have lower amounts coming out. But what I end up is a solute concentration in my tissues, so low at the top, high at the bottom. And I'm getting lower and lower solute concentrations inside the tube as well as I move up the ascending limb. So that's how we set up the actual solute concentration. And then water helps to maintain this by what happens on the descending side of the nephron loop. So on the descending side, when my filtrate first enters the nephron loop, I have a lot of water and a lot of solute. this number is actually so low. Even though I have a lot of solute, this number is small because I have so much water. And remember, these numbers are concentrations, and concentrations are taking into account both the solute and the volume of water. So even though I say high solute up here and I've got high solute down here, the difference in these numbers is because up here I've got lots and lots and lots and lots of water that is diluting that solute. However, water, remember, can come out of the descending limb, and water will always move by osmosis towards areas of more solute. So because there is more solute out here in the tissues now because of my action of my pumps, water is going to leave the descending limb to go out to where there's more solute, and so that's going to take away some of the water from the inside. So as I take away water from the inside, my concentration number is going to go up because I'm taking water away and making it more concentrated. Eventually, the inside concentration will match the outside concentration, but then as I move lower down the descending loop, my outside concentration goes up. So I have medium high water with high solute on the inside, but again, water is going to want to move toward an area of higher solute, so it's going to continue to come out, and I'm going to further concentrate what is left inside the descending limb as it moves down. So once it equalizes there, 600, 600 is equal, it's going to move down, but now I've got higher solute concentration out here again. So again, water is going to want to move out to equalize the concentration between the inside and outside. So notice that as I am coming down the descending limb, I am losing water, but my solute stays the same. So by the time I get down here to the very bottom, I have high solute but low water, and that's how I get this big number, and then the whole process starts over again. So this system works because of the different permeabilities. It works because the descending limb is only permeable to water, and the ascending limb is only permeable to solute. And the result, which is going to allow us to get concentrated urine, is a medullary osmotic gradient. So in the medulla, I end up with this concentration gradient with low solute concentration at the top and high solute concentration at the bottom. So the sodium and the chloride that is coming out in the ascending limb of the nephron loop helps with the concentration gradient, but urea also plays a role too. So urea is also going to contribute to the medullary osmotic gradient. And again, the medullary osmotic gradient is the gradient out here in the surrounding area that is surrounding the renal tubule and the collecting system. So the papillary duct, remember the DCT dumps into the collecting system, it dumps into a collecting duct, and then a bunch of collecting ducts come down into a papillary duct. Well, the papillary duct is permeable to urea. So urea can diffuse out at the bottom of the renal medulla, and that helps keep the bottom part very concentrated and helps keep the bottom of the renal medulla around 1200 milliosmoles per liter. So to summarize, the concentration of the tubular fluid when it enters the nephron loop is about 300 milliosmoles per liter. The concentration at the bottom of the nephron loop and out in the surrounding medulla is 1200 milliosmoles per liter. And the concentration at the top of the nephron loop and in the DCT is only 100 milliosmoles per liter. So now that gives us enough information to understand how we can produce concentrated urine. Also keep in mind, water is always moving by osmosis to an area with a higher concentration of solute. Water is also going to move through protein channels in the plasma membrane called aquaporins. So aquaporins allow very quick water movement across the cell membrane, which has that hydrophobic middle. And so whether or not a particular part of the nephron or collecting system is permeable to water is going to depend on whether there are aquaporins in the membrane or not. And when aquaporins are present, water will cross the membrane towards the area with the highest overall solute concentration. So urine volume and concentration can be regulated by controlling water reabsorption. So there are two major types of water reabsorption that takes place in the nephron and collecting system. And again, remember, water always moves by osmosis. So the first type is called obligatory water reabsorption. And it's obligatory because water reabsorption is always going to take place and the water permeability cannot be adjusted. So in other words, aquaporins are always present in the cell membranes in these areas. And so you have obligatory water reabsorption in the PCT, the proximal convoluted tubule, and in the descending limb of the nephron loop. So in these areas, you are going to always have water reabsorption. And in these areas, you're going to recover 85% of the volume of the filtrate, as we discussed before. Now, the other type of water reabsorption is facultative water reabsorption. So these are areas where the membranes are impermeable to water, except in the presence of antidiuretic hormone, or ADH. So the presence of ADH causes aquaporins to be inserted into the membranes of the cells, which allows for water to be reabsorbed. So it's facultative because it's going to depend on whether or not ADH is present. And this is going to take place in the distal convoluted tubule, or DCT, and the collecting system. The facultative water reabsorption provides control over the recovery of the remaining 15% of the filtrate volume. So remember, 85% is obligatory, it's always going to happen. And then how much we recover of the remaining 15% is going to depend on how much or how little ADH we have. The hypothalamus continually secretes low levels of ADH, so ADH levels can be quickly adjusted up or down as the body needs. And in the next couple of slides, we're going to look at the effects of ADH, but keep in mind, ADH is all about having the body retain fluid and retain water, and it is opposed by the natriuretic peptides, which want to promote fluid and water loss. So let's look at what happens in the presence of ADH. So remember, with ADH, we get aquaporins that are inserted into the cell membranes of the DCT and the collecting duct cells. So that means that with the aquaporins, the DCT and the collecting duct become permeable to water. So water will move out of the tubular fluid. Remember, we had a concentration of 100 milliosmolars up here in the DCT, but now water is going to move out of the tubular fluid and into the surrounding tissues. And remember, we set up that medullary osmotic gradient previously by using the nephron loop. So as we move down the collecting duct and the collecting system, and it gets more and more concentrated on the outside in the renal medulla, we're going to have more and more water get reabsorbed as that filtrate passes through the collecting system. So again, remember, we started with 100 milliosmolar per liter at the top. And so as we flow down the collecting duct, water is going to move out toward this area of higher solute. And remember, we had a concentration of 1200 down here at the bottom. So water will continue to flow out until what is inside the collecting duct matches what is the outside. So I'll end up with urine that could be as concentrated as the highest concentration at the bottom of the renal medulla, which is 1200 milliosmoles per liter. So this would be my most concentrated urine that I could produce. The actual final volume or concentration of the urine can depend on how much ADH you produce. So with more ADH, you'll have more aquaporins and you can reabsorb more water, but there are varying amounts of ADH that can be secreted. But if you had the maximum amount of ADH, the urine would eventually have the highest concentration, which would be 1200 milliosmolars per liter. So with ADH, big take-home message, with ADH, the kidney is going to produce a small volume of very concentrated urine. So now let's look at the absence of ADH. So if you have little to no ADH, you are not going to have aquaporins inserted into the DCT and collecting duct cells, so no more water would be absorbed. So urine would end up having the same concentration as the concentration of the filtrate in the DCT. And remember, that was 100 milliosmolars per liter. So again, we have this concentration gradient out in the medulla, but if we don't have any ADH, we don't have any aquaporins that have been inserted. Without the aquaporins, water cannot be reabsorbed. So what you had at the top of the DCT ends up passing all the way down, and that is the concentration of the urine you secrete. So without ADH, the kidney would produce a large volume of very dilute urine. And this is what happens in diabetes insipidus. Reminder, diabetes insipidus is the inability to produce ADH, and people who suffer from this can urinate as much as 24 liters per day. So that means they have the inability to do this facultative water reabsorption. So now we will do a summary of renal function and then briefly talk about urine analysis. So a summary of a lot of the principles that we've been talking about. Remember that the hydrostatic pressure at the glomerulus forces water and small solutes into the glomerular capsule to produce filtrate. The proximal convoluted tubule primary function is reabsorption. So it reabsorbs 60 to 70% of the filtrate, including water, ions, and nutrients. The nephron loop is going to reabsorb more water and ions, and it also helps to set up a concentration gradient through the countercurrent multiplication. The distal convoluted tubule is going to secrete additional waste products. The DCT and the collecting system also will make final adjustments in the volume, so they'll change the amount of water reabsorbed and the osmotic concentration through the reabsorption of ions. And these processes are highly regulated by antidiuretic hormone and aldosterone. And then the capillaries, the peritubular capillaries and the vasa recta, reabsorb water and solutes that were removed from the filtrate. So here is an overall summary picture of everything that we've been talking about going on at the nephron and the collecting system. So we started with filtrate being produced by the renal corpuscle. That filtrate then enters the proximal convoluted tubule, where you're going to have a lot of reabsorption. So reabsorption of nutrients, ions, and water. So between 60 to 70% of the filtrate is reabsorbed in the PCT. Then we have the descending loop of the nephron loop, and this is where you're going to have additional water reabsorption occur, and this is going to set up the concentration gradient of an increasing osmolarity in the tissues of the renal medulla. Then you're going to have the ascending limb of the nephron loop, where you have active pumps to pump out sodium chloride, which again is going to be part of setting up this concentration gradient in the renal medulla. Then in the DCT, you can have further adjustments in the composition of the tubular fluid, and this is going to be under the control of aldosterone. So the little A here in the circle, these represent sodium pumps that are controlled by the presence of aldosterone. You also have ADH, which can determine how many aquaporins are put into the cell membranes, and that is going to control water reabsorption in these areas. And then you also have your peritubular capillaries and your vasa recta, which are going to be running alongside the areas of the nephron so that they can reabsorb the nutrients, the ions, and the water and return them to the bloodstream. And then whatever you have left at the end of the papillary duct is going to become your urine. And then I'm just going to give you an overview into how we can test kidney function. I'm not going to go into the level of detail that your textbook does, but you should know this terminology. So a urinalysis is a chemical and physical analysis of a urine sample, and it involves kind of 3 stages. There can be macroscopic examination where you're just looking at the cloudiness and the color. There can be a chemical test. They have little test strips that you can use, and then you can also have microscopic visualization of the urine to look for things like cells or bacteria. So when you're doing the macroscopic examination of a urine sample, you're looking at the color and clarity of the urine, and the yellow color of our urine comes from urobilin. Urobilin is that molecule that comes from the breakdown of bilirubin, and if you remember, bilirubin comes from the breakdown of the heme group in the hemoglobin of our red blood cells. So whether your urine is pale yellow, borderline colorless, or dark yellow depends on the concentration and the volume of the urine. So the lighter the color of the urine, the more dilute your urine is. And the darker the color, the more concentrated the urine is, which could signal dehydration. Now there are, and so that's shown up here in the upper left of this picture, so well hydrated would be a very light color, whereas a darker yellow color would mean you are dehydrated. Now there are some other colors that you can get in your urines that can indicate various conditions. So I'll put this little chart here just to give you an idea of some of the other urine colors you can end up having. And then also keep in mind that medications and vitamins, especially vitamin B2, which is called riboflavin, can also change the urine color. So creatinine clearance, we mentioned before, this is going to compare the creatinine level in the blood with the creatinine level in the urine to estimate the glomerular filtration rate, or GFR. So normal GFR can vary by age. So as you get older, your GFR does decrease. In fact, between the ages of 25 and 85, you lose 30 to 40% of all of your kidney nephrons. So you have a slow decline in kidney function. But typically, a normal GFR is between 90 and 120 milliliters per minute. Another test is called a BUN, or a blood urea nitrogen test. This measures the amount of urea in the blood. So remember, urea is one of those waste products that we want to take out of the blood and put into the urine. So if you have high levels of urea remaining behind in your bloodstream, that indicates that you have a problem with your kidney function. So renal failure occurs when the kidneys can no longer adequately filter waste from the blood or maintain homeostasis. And since the kidneys can't filter the blood, we have to actually use machines to do it, and that is called hemodialysis, or just dialysis for short. So there is acute renal failure, which is a temporary condition that can happen because of renal ischemia, so that's blockage of blood flow to the kidney, urinary obstruction, trauma, or certain drugs. And then there is chronic renal failure, where you have damage that is usually irreversible to the kidney, and this can require long-term regular hemodialysis and ultimately a kidney transplant. So far we've mainly just talked about the kidney, but luckily the rest of the urinary system won't take as long to talk about, and we can cover it all in one section. So in this section we're going to look at the urinary tract, which includes the ureters, the bladder, and the urethra. So the urinary tract is responsible for transporting, storing, and eliminating urine. And it is made up of the ureters, the urinary bladder, and the urethra. It is lined by transitional epithelium, which is the type of epithelium that can stretch and then recoil back to its original position without accumulating damage. A pyelogram allows us to get an image of the urinary system using an X-ray and an intravenous injection of a radio-opaque dye. So the dye would be injected, and then you would have an X-ray taken, and then this would allow you to visualize the urinary system And in this picture from your textbook, you can even see the difference between the minor calyces and the major calyx within the kidney. So we'll start with the ureters. The ureters are muscular tubes that function to transport urine from the kidneys to the bladder, which is a distance of about 30 centimeters or 12 inches. The ureters are retroperitoneal, meaning they lie behind the parietal peritoneal membrane. And there is a slightly different path in men versus women due to the presence of the uterus. So the ureters start at the renal pelvis of the kidney, and then in men they can go straight down to the posterior side of the bladder. In women they have to go around the uterus, which sits on top of the bladder. The ureters then come in and penetrate the posterior wall of the urinary bladder. And where the ureters come into the urinary bladder, there are slit-like openings that make up the ureteric orifices, and this helps to prevent the backflow of urine back up into the ureters when the bladder contracts. The walls of the ureters contain smooth muscle, and there are waves of peristalsis that pass through the ureters. So the peristaltic contractions occur approximately every 30 seconds. And this is the same peristalsis that we talked about in the digestive system, but this is happening in the ureters. And it starts at the renal pelvis, and the muscular contractions move down the ureter to force urine towards the bladder. So the next structure is the urinary bladder. This is a hollow muscular organ that functions to temporarily store urine. So in this picture from your textbook, you can see the ureteric orifices that we mentioned. So this is low on the posterior side of the bladder, and this is where the ureters will actually insert and deliver the urine. So the urinary bladder is stabilized within the abdominal pelvic cavity by the umbilical ligaments, and there is a median umbilical ligament and two lateral umbilical ligaments, and these are vestiges of the umbilical arteries that we talked about in an earlier chapter. The mucosa, which lines the urinary bladder, has rugae, just like the stomach does. And so these ridges allow for expansion of the bladder as it fills with urine. And then at the bottom of the bladder is a trigone, which is a triangular shaped smooth region that is bounded by the openings of the ureters and then the urethra at the bottom. So think of it as having three corners. The two corners at the top are the two ureteric orifices, and the corner at the bottom would be the opening to the urethra. So here's another image that can help you visualize the trigone, and you can see the ureter openings, and then down here would be the opening into the urethra. So the trigone has a very smooth and very thick mucosa, and so it acts as a funnel to channel urine into the urethra when the bladder contracts. There is an internal urethral sphincter, which is located around the neck of the urethral opening right at the base of the bladder, and this sphincter is under involuntary control. And then there is an external urethral sphincter, which is a skeletal muscle band in the perineal muscle of the pelvic floor, and this is the one that you have voluntary control over, but you don't get that voluntary control until around the age of 2, and so that's when you start being potty trained and learning how to control that muscle. So there are stretch receptors in the bladder wall, and you get the urge to urinate when the volume inside the bladder reaches 200 milliliters, although some people are able to hold their urge to urinate for a long period of time, and it has been known for the bladder to hold up to a liter of urine. After urination, only 10 milliliters remains behind. The detrusor is a powerful muscle that is within the wall of the bladder, and the detrusor muscle contracts during the process of urination. There are two reflexes covered in your textbook, but I'm not going to go into the level of detail your textbook does. They involve a complex interplay between sympathetic and parasympathetic innervation. But there is one reflex called the urine storage reflex, which allows you to continue to store the urine in your bladder when you decide you're not going to quite go to the bathroom yet. And then you have the urine voiding reflex that kicks into play when you do go to the bathroom and you've decided to let your urine out. Incontinence is the term we use to describe the inability to control urination voluntarily. This can happen from damage to either of those sphincters. It can also happen with the loss of muscle tone in those sphincters that occurs with age. And then you can have damage to the CNS, either in the brain or the spinal cord, which can also lead to incontinence. So renal calculi is the official term for a kidney stone, and these form within the urinary tract from calcium deposits, magnesium salts, or uric acid crystals that basically come together and conglomerate into a stone. So nephrolithiasis is the condition of having kidney stones, and if you break this down into the root words, nephro meaning having to do with the kidney, and lith, if you remember from earlier in the semester, means stone, so this is the condition of having kidney stones. And if a stone ends up blocking one of the ureters, this is called a urinary obstruction, and if it's a complete blockage, it can be an emergency situation. It can cause extreme pain and can also increase the hydrostatic pressure in the glomerular capsule, which can interfere with filtration of the blood. Small stones that don't cause a complete blockage can be passed naturally. However, these can be very painful, and they cause waves of pain that occur with those peristaltic contractions in the ureters. So you typically feel the waves of pain come and go like about every 30 seconds, and so that coincides with the peristaltic contractions. Larger stones that can't be passed with the fluid pressure and the peristalsis can be surgically removed or broken up using sound waves or lasers. The best thing you can do to prevent kidney stones is just to drink more water. The more dilute your urine is, the less likely those salts and other compounds can come together to crystallize and become stones in the first place. So the more concentrated your urine is, the less water you have, and the higher solute concentration, the more likely you are to develop stones. And then the last structure is the urethra, which transports urine from the bladder to the exterior of the body. So the urethra is different in males and females. So if we look at males first, the urethra transports urine and sperm in males. And because it has a much longer distance to travel to get from the bladder to the exterior of the body, the urethra averages 7 to 8 inches in males. And for this reason, it is divided into 3 segments. The first segment is the prostatic urethra, which passes through the center of the prostate gland, and we'll talk more about the prostate gland in chapter 28. Then you have the membranous urethra, which passes through the muscles of the pelvic floor. And then you have the spongy urethra, which passes through the penis. The external urethra orifice in males is at the tip of the penis, so that is where the urine will come out. In females, the urethra transports urine only, and it has a much shorter distance to travel to get from the bladder to the exterior, so in females it is only about 1 to 2 inches long. And the external urethra orifice in females is in a structure called the vestibule, and it sits anterior or just in front of the opening to the vagina. So our last section will kind of be a review. We'll talk about the excretory system, and it will allow us to review several things that we have covered in several chapters. So the excretory system is comprised of components from multiple organ systems that all work to get rid of waste, so to get waste out of the body. So we've talked about the urinary system in this chapter, and the urinary system gets rid of organic waste products like urea, creatinine, and uric acid. We also didn't really go into much detail with the integumentary system, but the integumentary system is going to be like your skin and sweat glands, and that can eliminate small amounts of metabolic waste in your perspiration, which is your sweat. We did recently talk about the respiratory system, which is a key system for eliminating carbon dioxide. And then the digestive system, we also recently talked about. The liver can excrete waste products into the bile, and then the digestive tract eliminates solid waste through the feces. And that is the end of chapter 26.